Hi, this is Virginia, and this presentation is called Genetically Modified Foods, Background and Controversy. The purpose of this presentation is to explain how we get genetically modified foods and go over some of the controversial issues surrounding genetically modified foods, but this presentation doesn't touch on every single controversial part of this issue. So, creating genetically modified foods involves tinkering with nature, which is something that has happened for a very long time in agriculture. And there are ways to tinker with nature without using biotechnology. Farmers and other plant breeders have tools to create crops that have many desirable characteristics and few undesirable characteristics. And for the most parts, these kinds of tinkering have increased yield or better quality crops. Some of the characteristics that farmers breed for are bigger flowers, better taste of the vegetables or fruits that they're growing, richer color, increased tolerance to heat, cold, or drought. So like I said, farmers have been doing this for a very, very long time. And they use three processes. Selective breeding is the first process. Selective breeding is where you choose to plant seeds from plants who have the desirable characteristics that you want. So you can see that the wild mustard plant has been selectively bred to create all of these different plants like kale and broccoli and cabbage. And say to produce kale, the farmer, over a series of many generations of the plant, would keep selecting wild mustard plants that have these long leafy greens, and then they would keep selecting them and planting those seeds, and eventually they would start to create a new plant called kale that had this trait that they had been selecting for. The second process that farmers have been using for a long time is called hybridization. Hybridization is when you cross two genetically different individuals to result in a third individual with a different set of traits. So plants of the same species cross really easily, but when you're crossing plants of different species, it can get a little bit more difficult. And oftentimes the result of crossing two different species is sterile and they don't produce seeds. So this is an example of a long, thinner yellow pepper and a shorter red pepper. And in hybridization, you would take pollen from the yellow species and use that to pollinate the red species, and then the hybrid of those two is a longer red pepper that has traits of both of its parents. But this longer red pepper may not have seeds that will actually grow into the long red pepper. Those seeds might not grow. So oftentimes with hybridization, you have to recross-pollinate with every single generation. Grafting is the third process that farmers have been using for a long time. And when you graft, you take tissues from one plant and insert them onto the tissues of another plant. And what you're doing is you're getting the vascular tissues of both of those plants to join together. And in most cases, one plant is selected for its roots and the other plant is selected for its stems, leaves, and flowers. And one of the reasons people do this is to create dwarf plants. For example, in modern orchards, apple orchards, you would graft a full-sized apple tree onto a dwarf stump. And this would allow you to have the yield of a full apple tree at a very low to the ground level, so that it's easier to pick the apples and that you don't have a lot of injuries with apple pickers falling off of ladders and things like that. Another reason that people graft is to shorten the juvenile stage of a plant's life cycle. The juvenile stage is a natural stage through which a seedling plant must pass through before it can become reproductive. And in most fruiting trees, the juvenile stage lasts between 5 and 9 years. However, you can shorten the juvenile stage by grafting mature plants onto young rootstocks. So in this photograph, you see plants, you see the branches of plants that are already mature and are able to produce fruit, and they're grafted onto a younger rootstock and trunk. So this plant will start producing fruit again much earlier than the plant would have originally produced fruit. Now we're going to talk about ways that scientists have changed plants and agriculture. And these techniques cannot be done in a field, they have to be done in a laboratory. So this is an example of a scientist getting some genetically modified plants started in a lab. So the technology that scientists use is called recombinant DNA. And how we're going to do this is we start with a bacterium called Bacillus thuringiensis, which produces a protein called Bt toxin. And Bt toxin acts as an insecticide, so it kills insects, but it does not act on humans or other animals. And we're used to thinking about DNA as long strands wound up in chromosomes, but bacteria house their DNA differently. Bacteria have their DNA in these rings called plasmids, and on the screen you can see these rings, and that is where their DNA is. So we want to take the Bt toxin gene out of the Bacillus thuringiensis bacteria, and we want to get it into the plant. So we're going to use enzymes to cut the plasmid, cut out the gene, and then insert the gene into a different bacteria. And the second bacteria is called Agrobacterium tumefaciens. So Bacillus thuringiensis had the gene we wanted, but Agrobacterium tumefaciens has the unique ability to act on plants at the site of an injury and insert its plasmids into plant cells. So in this diagram, we have Agrobacterium tumefaciens, and we're going to cut it where it says site of cleavage. Cleavage means that's where we cut the plasmid. And then we take these specified genes, which in this case are the genes that code for the production of Bt toxin, and we're going to get those genes to stick to the sticky ends of the plasmid. And then we seal the deal through annealing the plasmid back together. So now we have the Bt toxin genes inside the Agrobacterium tumefaciens. And then the Agrobacterium tumefaciens is going to act on the plant. So you need to injure the plant and then smear some Agrobacterium tumefaciens on the injury, and at the point of that injury, the Agrobacterium tumefaciens is going to transfer its DNA into the plant. So that is one method of getting this bacterial gene into the plant's DNA, but there are a couple of other methods. You can use a very tiny gun, and you take the Bt toxin gene and you coat it in gold or tungsten, and you use this little tiny gun that's powered by helium gas, and you shoot the DNA into the plant cell. And a third method of getting DNA into a plant cell that didn't originally belong there is by shocking the cell. And when you shock the cell, the pores of the cell open temporarily and they allow the DNA to pass through. So ultimately, you're ending up with a plant that kills the insects that feed on it. So in this diagram, you can see these plants are taking up the insecticide gene. You're making sure some of the plants aren't going to take up the gene, so you're going to get rid of them and you're only going to keep the ones that do take up the gene. And then you're going to grow those plants, and then the insects that feed on them will die. Aside from making plants resistant to insects, genetic engineers can also make plants resistant to herbicides. Glyphosate-based herbicides like Roundup all work on the same biochemical principle. They inhibit a specific enzyme that plants need in order to grow. The specific enzyme is called EPSP synthase, and without that enzyme, plants are unable to produce proteins that they need to grow, so they yellow and die over the course of a few days or weeks. The majority of plants all use this same enzyme to grow, so almost all plants succumb to Roundup. Plants that are modified to be resistant to glyphosate-based herbicides use a different enzyme to grow, so they're not affected by the inhibition of the EPSP synthase. So you can spray these crops with herbicides, but the crops won't die. Everything else will die because these crops are resistant to the herbicide. In addition to making plants herbicide and pest resistant, plants have been developed that can endure cold temperatures or drought. They can resist fungus, they increase crop yield, they make more nutrients than the conventional plants would, or you can even grow plants that produce vaccines or drugs. So that was an overview of how we got to genetically modified crops, from selective breeding, hybridization, and grafting, all the way to using science and laboratory techniques to get bacterial genes that would never find their way into plants. And now we're going to talk about some of the controversies surrounding these processes. A lot of people are worried about potential human health concerns. So every single genetically modified crop goes through an approval process with the FDA. And the FDA looks at all of the scientific research surrounding the crop and the food, and they look at all of the evidence, and they either reject or approve each individual species. So many species have been rejected, some have been approved. And this is a fairly rigorous process, although not everyone trusts this process, which is why, there are still people who fear that there may be health effects associated with GMOs. And one of these health effects that people are worried about is cancer, because there was a French study that found cancer in rats that ate a genetically modified diet of corn. And the researcher, Gilles Eric Seralini, is a molecular biologist who published a study this year. And he did an experiment where scientists fed 18 different groups of rats various concentrations of genetically engineered corn or the herbicide Roundup. So they had 200 rats, and they split them into 20 groups, and each group had 10 rats in it. They kept 2 of these groups as controls, so the controls ate not genetically modified corn, and then the 18 other groups ate genetically modified corn, or they had the herbicide Roundup mixed into their feed. The experiment went on for 2 years, and on average, the rats that ate genetically modified corn or drank water that was laced with Roundup did worse than the control group. They developed tumors that showed up earlier in their lives, and more rats died in the experimental groups than in the control groups. The issue with this study is that it has been critiqued by many scientists. And there are a lot of questions regarding the study. One of the questions is of the researcher's own bias. The researcher, Gilles Eric Seralini, has been campaigning against GMOs since 1997. He sells books about how GMOs are dangerous, and he has a clear interest in proving that they're dangerous. So he walks into this study as a very biased person, and there are ways that you can perform research that is biased. So scientists are concerned that his research is biased.